Thank you very much, Saya, for that great presentation. So I'm Maria, I'm Senior Scientist here at IBL in Cardiff, and I've been overseeing the development of our simple T-cell test. Since the start of the pandemic, there's been a lot of research done investigating the immune responses to SARS-CoV-2. Initial studies revealed that virus-specific T-cell responses developed in nearly all individuals with confirmed SARS-CoV-2 infection, and that these SARS-CoV-2 reactive T-cells were capable of co-producing multiple cytokines, such as interferon gamma, IL-2, and TNF-alpha. From studies that have been done on closely related MERS and SARS-CoV-1 viral infections, it was predicted that these T-cell responses would be enduring. And in fact, this has been shown to be true, with papers published earlier this year that have demonstrated the persistence of SARS-CoV-2 T-cell responses up to at least 8 months post-infection. Next slide. So early in the pandemic, multiple commercial antibodies were rapidly developed and became commercially available. But there were some questions around their sensitivity and specificity. Additionally, there's been research articles that started to come out that reported antibody titers were declining quite quickly over time. And as Saya's data showed, this is certainly in agreement with this. Therefore, perhaps measuring T-cell responses could potentially be a better readout for population immunity. The problem is that traditional assays that are used to measure antigen-specific T-cell responses, and these are things such as ELISA or flow cytometry, these require separation of the T cells or the white blood cells from the whole blood first, before you can even begin the assay. And consequently, these techniques can be labour intensive and require specialist technical knowledge. Therefore, with funding that we got here at IBL last summer from Innovate UK, our aim was to develop a rapid, simplified test to measure virus-specific T-cell responses in whole blood. Next slide, please. How the assay works is that, firstly, venous blood is drawn into a vacutainer, and this contains sodium heparin. This is an important step because it stops the blood from clotting, but it doesn't cause T-cell activation, which things such as EDTA are known to do. The blood is then transported to our laboratory here in Cardiff, and we split it into 3 different tubes for 3 different stimulations. There's one tube that contains PBS only, and this is our negative control. One tube that contains PHA, and this acts as our positive control for the assay, as this causes pan T-cell stimulation. And then we have our test tube, which contains the megapool of SARS-CoV-2 peptides. And this megapool consists of 470 overlapping peptides, which span the entire immunodominant regions of the SARS-CoV-2 proteome. Samples are then incubated overnight, the plasmas are collected, and then we perform the immunoassays to measure cytokine production. Next slide, please. So firstly, interferon gamma. I'm just going to show you an example of a positive and negative donor. The first graph on the left is an example of what we get when we stimulate the blood from a PCR-confirmed positive donor. So the negative sample, which is a circle shown in grey, was below our cutoff of 24 picogram per mil, as we'd expected. And our positive sample in green, as well as the test sample, the SARS-CoV-2 peptide stimulated sample in blue, are both well above that cutoff. The middle graph then is from a PCR negative individual. Again, the negative control is below the cutoff, as we'd expect. The positive sample is well above the cutoff, as we'd expect, but this time the SARS-CoV-2 peptide stimulated sample was also below the cutoff, clearly showing the difference between positive and negative donors. So the graph on the right then is basically the summary of all 128 individuals that we recruited last year between June and December. The cohort shown in the red circles are those that have received a positive PCR test result previously. The blue cohort are those with what we've termed suspected infection or exposure. However, they did not have PCR test probably due to the slower rollout of routine testing, especially at the beginning of the pandemic. And then we have the green cohort, who are individuals with no known history of exposure. In the PCR positive cohort, we can see that 97% of individuals demonstrated a positive response, along with 80% positivity rate in those with suspected infection. And this is in comparison to just 20% of donors from the cohort of unexposed individuals, who also demonstrated a much lower magnitude of response. Next slide, please. So the results from Gamma demonstrated to us that this whole blood approach was definitely a viable one. So we next investigated if there were any other cytokines that could also distinguish between T-cell responses from natural infection, as opposed to those individuals that had not been affected. We found that interleukin-2 was actually an even better, more accurate biomarker, as demonstrated in the graph. There's a clear distinction in the magnitude of IL-2 responses between individuals with previous PCR infection, again shown in the red dots, compared to those in the green with no known history of exposure. And as you can see from the coloured box, the mean IL-2 levels rose from just 9 picogram per mil in those with no known history of exposure, all the way up to a mean of 328 picogram per mil in those with PCR confirmed infection. And I'd just like to point out that this time all 29 PCR individuals demonstrated a positive IL-2 response above the cutoff of 20 picogram per mil. And this is in marked contrast to just 1 out of 69 from the unexposed cohort that you can see. Next slide, please. So we also analysed multiple other cytokines, and out of 15 others that we assessed, only IL-13 and IL-6 showed any significant differences between the PCR positive and the uninfected groups. But neither are as accurate as IL-2 or even interferon gamma at differentiating between those cohorts. The magnitude of IL-13 response with most samples is very small, as you can see, of less than 10 picogram per mil. And a lot of the results were actually below the limit of detection for the assay. And as for IL-6, despite the significant stars on the graph, there was actually no clear distinction between the infected and the uninfected individuals, as there's just far too much overlap. Next slide, please. So to analyse the results a little more, receiver operating characteristic curves were generated for both IL-2 and interferon gamma. Now, these curves provide an indication of both sensitivity and specificity of your diagnostic test, and they produce a value known as the area under the curve value, or the AUC value. And what you want is basically this number to be as close to 1 as possible. The closer that it is to 1, the more accurate your diagnostic test is. And although both AUC values for IL-2 and interferon gamma are extremely high, the AUC value of 0.9950 for IL-2 was closer to 1 than the 0.9465 value of interferon gamma. And this just really emphasises again that IL-2 is actually the more accurate of the two as a biomarker. Next slide, please. To conclude this section, for natural infection, the rapid whole blood test can distinguish SARS-CoV-2 infected individuals from uninfected ones, and the magnitude of IL-2 and interferon gamma responses increases 36-fold and 11-fold respectively from previously uninfected individuals compared to those infected individuals. Next slide, please. So with the rollout of the global vaccination programme, we next addressed the question of if our whole blood test could detect vaccine-induced immune responses as well. So to do this, we took 2 cohorts of individuals, this time one from the UK and one from the US, and the assay setup was done in exactly the same way as before. Next slide, please. So the participants for the vaccine work were divided up into those that had no previous history of infection, versus those that had confirmed PCR for previous exposure. Graph A firstly shows results from the previously uninfected individuals. There was an 18-fold increase in magnitude of IL-2 responses between pre and post-vaccination. The pre-vaccination responses are shown in red, and the post-vaccination responses are in blue after the first dose and green after the second dose. We can see that following vaccination, 97% of individuals demonstrated a positive IL-2 response. Secondly, in graph B, this shows data from those individuals with a previous PCR-confirmed infection. All donors demonstrated a positive IL-2 response following the first and second doses of vaccine. But interestingly, vaccination did not boost the mean of the IL-2 response to above those seen pre-vaccination. Next slide, please. Again, we analysed multiple cytokines in case we missed any other important ones, and actually only 3 others showed significant differences again. As expected, we did see great response to interferon gamma again. Mean interferon gamma levels, which are shown in graph A, increased by approximately 10-fold following vaccination. Although, as with the natural infection graph, the distinction between the groups isn't quite as clear-cut with IL-2. With IL-13 shown in graph B, the magnitude, as with natural infection, was again very small, and there were some vaccinated donors who did not produce a positive IL-13 response at all. And with IL-10, the magnitude, again here, very small, and although there was a significant difference between the pre-vaccinated samples and samples taken after the second vaccine dose only, as with interferon gamma and IL-13, there was again quite a lot of overlap between the cohorts. So we can't really say again that IL-13 and IL-10 are particularly good biomarkers. Next slide, please. Okay, to conclude the work from the vaccine-induced responses, the mean IL-2 response of previously uninfected individuals increased by 18-fold after vaccination, with 97% of those individuals demonstrating a positive vaccine-induced IL-2 response. Consequently, our conclusion is that IL-2 is a highly accurate biomarker for distinguishing unvaccinated and vaccine-induced T-cell responses in our whole blood test. Next slide, please. For my final couple of slides, I just want to discuss the next steps for the development of this test. Although our results are extremely promising, the test is not yet fully validated or ready for commercialization. However, I'm really pleased to announce that we've just been awarded a new Small Business Research Initiative grant from Innovate UK to further develop it. So over the next 12 months, we have 4 major milestones which we wish to achieve through this grant. The first 2 are being focused mostly on method validation and development, and I won't go into those here. The third milestone, however, is really where the new innovation lies in this test, and this is going to involve establishing a minimum volume of blood that we can still yield a measurable amount of cytokine from. And the reason for this is if we can get down low enough, the aim is to compare responses from venous blood draw with responses from capillary blood draw, with the main goal of this being so that we can make this test a far more accessible one, as it would no longer require the need for a trained phlebotomist to be present to take the venous blood. Next slide, please. So milestone 4 is going to be done in conjunction with some amazing collaborators that we have from the NHS South Tees Hospital Trust. And with their help, we're going to put this test into a clinical setting where we hope to address the critical question of if there are differences in magnitude and longevity of vaccine-induced T-cell responses in clinically vulnerable individuals, or those individuals that are deemed more at risk from infection than immunocompetent individuals from the wider population. This work will be monitored by the immunity arm of the National Core Studies programme, which has been set up by the UK government. And we really hope that the information gathered by us over the next year will help aid decision-making on public health policies and vaccine efficacy. Next slide, please. So that just leaves me to acknowledge all the fantastic people who have helped with this work, specifically everybody here at IBL in Cardiff, who are absolutely fantastic team to work with. Then we have to thank Dr. Martin Chapman, Dr. Nicole Brackett and Brian Smith from Biotechnologies Inc. in Charlottesville. Dr. Hannah Hoff, who did a tremendous job in taking blood from multiple donors for both the natural infection work and the vaccine-induced work. And then Dr. David Chadwick and team, who are our amazing collaborators from the South East Hospitals NHS Foundation Trust. And also then a massive thank you to Innovate UK and SBRI, who have funded this work and funding the work to be done in the next 12 months.